time to pick another number from the hat. Here we are, number, drum roll please, 6. Number 6. Or is it a 9? It's a 6, I'm pretty sure. So, number 6, we'll go to our slide case. And we are looking at lower epiderm of leaf of horsebean. The lower epiderm of leaf of horsebean. So we'll take a good look at the lower epiderm of leaf of horsebean. Welcome Zorrocats to 200,000 times closer, where today we're looking at the horsebean leaf, which is more commonly known as the fava bean. The fava bean goes great with a nice Chianti. Well, that's beside the point. We're going to be speaking a lot today regarding the stoma, which allows gas transfer for these plants. The horsebean is just a great example of how most plants function in allowing gas in through the lower side of the leaf. The lower side of the leaf is filled with pores called stomata. The stomata are simply gateways guarded by guard cells. And during the daytime, the guard cells open up due to, well, we'll get to that. It's kind of like a bouncy castle where they're filled with pressure, and in so opening the passageway for gas to be let into a spongy layer of the leaf. The spongy layer contains all of the living cells, as well as some dead cells that continue to provide structure and support for the leaf. But most of the living cells are on the outside layers to gain sunlight for photosynthesis. Photosynthesis, of course, creates the energy for the plant to allow turgor pressure. And turgor pressure opens guard cells. It also provides water to the leaves and through the stem. And in this case, these pores are closed at nighttime. The sunlight provides the plant with energy. Photosynthesis creates this pump. It opens up by putting out hydrogen ions and allowing potassium to fill these guard cells. So let's take a look at this in the microscope here. Now, I also have a cheat sheet, so don't mind me here. The guard cells, when swollen, are turgid. When they're shrunken or flaccid, they're closed, and you can see then the chloroplasts and the vacuoles, the cell wall and the nucleus and all of that. We're going to get in close here. The leaf, you can see the squiggly lines. That is the epidermis cell. The many pores are the stomata, plural, of course, of stoma is stomata, the pores, which allow gas to reside inside of the spongy layer. Now, it's very passive as far as a transaction. The plant in general is a very, I would say, passive plant. And in saying this, I'm here in the dawn of spring, and there's a common misconception that these plants and trees, they die in the winter, and it's furthest from the truth, because they're simply letting go of what is not needed and becoming tough and strong through the cold winter months where they're still very much alive. And these plants, even perennial flowers, have a bulb where they cut off energy to the non-essential sections of the plant in order to kind of hibernate and survive through those cold winter months. They're not at all dead. The leaves die, cells die. It's part of life where we have a cycle of creation and therein the death of these cells. But also it's most interesting to me that the dead cells providing structure, and if you want to say a skeleton for the plant, are very much like our hair and fingernails, and the dead cells that we have that also do the same. They still provide function within living organisms. So The epidermis has dead cells. The spongy layer, plasma layers have dead cells that provide the structure. In the spongy layer, it's very open to airflow because gas transfer happens much more quickly, a multiple of 1,000 times quicker in open air than in liquid. So in plants, although they have a very functional liquid transfer system with turgor pressure, they still rely on open air transaction for the transpired air. So it is allowed to contain carbon dioxide. Photosynthesis releases the oxygen once carbon is taken, combined with hydrogen dioxide, which is water, and you have carbohydrates, which are sugars. And of course the excess is many molecules of oxygen. So the oxygen is the transpired air, which is allowed to pass through these open stomata, plural. So we're looking through our microscope at this lower leaf section, and I can see it's very, very prevalent for these stoma to provide that gas transfer to many areas of the plant, whether it's in the stem, the lower side of the leaf, the spongy layer through to the living cells at the top of the leaf as well, where it's gaining light, the blue light. And I just want to get in close, get in focus, and here we can see the stoma and the guard cells. They look similar to lips, if you would. These lips, when inflated, are open. When they're deflated, are closed. And I used the analogy of a bouncy castle because it is very similar, with the pressure inside pushing the outside wall to create a space. There we have it. Okay? So currently, this is 10 times magnification, and I think my eyes are fooling me. There we are. 10-time magnification of the bottom of this fava bean leaf. But this is on all types of plants, for your maple trees, oak trees, flowers. They have the same stoma or stomata system for gas transfer. Now we're going in again, and here we have a great view of a few stoma, a few stomata. And to explain it perhaps a little better, to describe the process a bit better, in the daytime, during the blue light absorption, phototropin activates a proton pump. The proton pump for these guard cells simply means that the guard cell contains hydrogen positive ions, which are put out through osmosis, and in doing so they fill with potassium and they expand on the outer walls, allowing that opening to form. And this is how we transfer gas in plant life. If you ever think that you fully understand something, there is so much more to learn. And I look into something like a leaf, and I think that there's not much that I could take from it. And I'm always surprised with how many questions come from learning about the simplest, or I wouldn't say the simplest, because you could spend a lifetime just focusing on one aspect of a plant. And even something so passive as a pore that opens for carbon dioxide and oxygen to be exchanged, there's a chemical curiosity that I have so many questions about. The hydrogen ions being pushed through osmosis out, and the potassium and filling these guard cells. Where does the potassium come from? The hydrogen exists in there naturally, or I'm just confused there personally, but still nonetheless. a simple leaf has really gotten my curiosity piqued. So with that being said, we talk a lot about photosynthesis on the show. I feel that we're going to have to eventually have an episode focusing specifically on the cell and how the plant itself sustains all of these different, what seem to be like, there's no brain, there's no system center. The roots, or you can consider it a system center, but it's not as clear as in an animal. The heart pumps the blood, which contains all of the oxygen. The plant doesn't do that. All of the cells are readily supplied with the water from the root, Tolger pressure pushing it upwards. The gas is exchanged right at the source, in the pores. So we have to get into eventually the cellular level, how these chemical reactions occur, how they send signals to one another, and this is just a great opening segment, I suppose, for all of that. And it's just opening curiosity. So we'll go in one more time here, and we'll take another look at the horsebean leaf, the epidermis cells, and we shall have questions that come from it, sometimes more so than answers. Quite simple, but dynamic. The squiggly lines of the epidermis cells. The guard cell, of course, is a cell in and of itself. Each end, I should say, it's a pairing, a partnership. But this partnership, they each have their own nucleus. They each have their own vacuoles and cell walls and chloroplasts. Raises so many questions. You know, it raises so many questions. The DNA of the plant, it has a lot of secrets that we should be interested to find out. And we are lucky here to have just the lower segment that I want to show you on this side here to give you an idea of what a side cross-section of the plant. So you can see the lower epidermis, which is what we have here. Then there's the plasma layer, a spongy layer, and the thicker upper epidermis, which has only protective functionality, I would say, as a curious man, not a well-educated man. The epidermis in general is coated with a waxy layer and is very rigid. The plant in and of itself, it's spongy internally and rigid externally for protection because it is subject to so many external forces and has such a passive lifestyle. And with that, it's fascinating that there are so many species of plant all over the world that exist due to evolutionary progress. Whether it's seeds that last the winter, or whether it's a bulb that exists underground, cutting off any non-essential life-sustaining elements, whether it's the tree that develops a hard, coarse bark and internally stores everything it needs throughout the winter. It's just a wonderful thing, and it's truly a blessing that these plants exist for our sake, and it's not just the beauty of it, but so we can breathe our oxygen and we have a partnership with them in everyday life. It's crucial for us to exist together, I would say, animals and plants. But that's where I have left us for today. If you have more questions or something to add to the conversation, I encourage you to do it in the comments below. We're all searching for answers, and I think together is the way to find them. Truly a blessing to be with you guys, and I thank you for watching every single time. It's humbling. The experience of learning is always humbling, and I thank you. If you thought this video was magnificent, then be sure to click the like button. You can always share. Be sure to subscribe by clicking the icon in the credits, and as always, my best to you.